Hello viewers, welcome to our online lecture. I am Dr. Archana Chaube from the Department of Chemistry, Bhopal School of Social Sciences. Today, we'll be starting with an interesting branch of chemistry, that is spectroscopy. Our today's topic is UV visible spectroscopy. So let us begin. First, we must understand what is the meaning of spectroscopy. Spectroscopy is actually a branch of science which deals the study of interaction of light with the molecules, and after this interaction, what are the changes that occur in the wavelength frequency of the light, which is being measured by specific instruments which are known as spectrophotometers. And when these interactions are studied, the results are interpreted in the form of graphs. So these graphs are known as spectrum. So spectroscopy is what? It is a branch of science where you are studying the interaction of light, and for which different instruments or spectrophotometers are used. Now, when I am talking about spectroscopy, broadly if we classify, spectroscopy is of two types depending on whether we are analyzing the light absorbed during the process or the light which is emitted out during the process. So, on that terms, the two spectroscopic broader classification are absorption spectroscopy and emission spectroscopy. Now, what is absorption and emission spectroscopy? If in this diagram we see that suppose electron is in the ground state, and when we radiate it with a desired amount of light radiation, it absorbs the radiation and it jumps from the ground state to any higher excited state. During this process, it has absorbed the light which has fallen on it, due to which the transition has occurred. And if in the phenomena we finally analyze how much amount of light has been absorbed for the process, such spectroscopy will be classified under absorption spectroscopy. But the other way around is also true, like after excitation, the process, the molecule comes down back to the ground state, for which there are various given steps. But during this process, since it comes down, it will radiate out the energy that it has already absorbed for the excitation. So, if we analyze this energy or the radiation which is being emitted out while returning back to the ground state, then the spectroscopy will be termed as emission spectroscopy. So, based on the type of radiation that we are analyzing, it can be classified as absorption spectroscopy and emission spectroscopy. Now, moving further, since I am talking about light radiation, so we must know the meaning of electromagnetic spectrum. Like we all know, light is a form of energy, and the formula for energy is E is equal to h nu in terms of frequency, or we may say E is equal to hc by lambda in terms of wavelength. We know that frequency and wavelength are two opposite terms. If frequency is increasing, the wavelength is decreasing, and vice versa is also true. So, what is the meaning of electromagnetic spectrum? If you look in the atmosphere, there is a vast range of radiations, energy radiations which are present. And here in the diagram, we may see the arrangement of these radiation, both in terms of, if we see in terms of increasing order of wavelength from left to right. For the ones which have smallest wavelength is the gamma rays, then X-rays, UV, visible, IR, in between comes the microwave, and then comes the radio waves. This is in terms of increasing order of wavelength. So, accordingly, these radiations have their use. Like for example, we know that radio waves are those waves which are used for telecommunication, in mobiles, and in our technology for TV transmission. These are the radio waves, because they are long waves, they can travel long distances. But in terms of energy, their energy is very low. And whereas on the left hand side, the strongest energy wavelengths are the gamma rays. And today we will be focusing on our spectroscopy that we have started with, that is the UV visible spectroscopy. Now, since there are different radiations and all radiations have the ability to cause excitation, but since we know that the energy of every radiation is not the same, so obviously the excitation that they are causing is also not the same. So, let us see what type of excitation each radiation may cause. Now, I have used a term dual nature. What do we mean by dual nature? The electromagnetic radiations are show dual nature. That means they have the properties of wave as well as particle. They move like waves. Whenever we show light radiation, we show the movement in terms of waves. That is why they have the property of wavelength as well as frequency. They also have the particle nature, and how it can be confirmed, because light radiation is made up of small packets, energy packets or quantas, and these are the packets or quantas, particles which are absorbed by any molecule, due to which the excitation occurs. Now, what are the uses of different light radiation? We have taken 4 important light radiations that are generally studied under spectroscopy. The UV visible radiation, it causes which transition? It causes electronic transition in the molecule. Then comes the IR radiation, which causes vibrational excitation. Then microwave radiations, which causes rotational changes in the molecule, and radio waves causes the reorientation of the nuclear spin. Like, because in terms of energy, radio waves are those which have least energy. So, when these radiations are irradiated, they do not cause much change in the bonding of the molecule. They only cause a change in the nuclear spin, and gradually as you move upwards, microwave is causing a change in the rotation of the molecule or the atom. The IR is causing still more change. Along with rotation, it is causing vibrational changes as well, and the UV visible has highest energy among the 4. So, this causes the electronic transition. That means the electrons jump from one level to another, causing the excitation. Now, in case of UV visible spectroscopy or any spectroscopy, we must first understand what is the principle and the instrument that is used for the study. So, if we talk about the principle of UV visible spectroscopy, it is based on the absorption of the ultraviolet radiation, that is the UV, and the visible radiations by compounds, and that produces different graphs, or which are known as spectra. So, when a matter absorbs ultraviolet radiation, the electrons, they undergo excitation and they jump from the ground state to the excited state. I have already discussed that, and this excitation will only occur if the energy gap which is there between the ground state and the excited state, the minimum amount of energy that is required for this transition is obtained, and the light has that much of capability to give that energy, then only the transition will occur. If you are radiating the molecule and it doesn't get that desired energy gap, and it doesn't get that amount of energy so that it can jump from the ground state to the excited state, the excitation will not be possible. So, we are saying that it is important to know the difference in the energy of the ground state and the excited state. It must be equal to the amount of the radiation that is being absorbed. Like, it is like this. Suppose this is the ground state, S0 state, and this is the next excited state, S1, and this the axis is showing the energy. So, there is some energy gap, delta E. Whatever is its value, if this amount of energy is given to the molecule, then only the excitation will be occurring. Now, all those molecules that have pi electron, or they are unsaturation, they have non-bonding electrons, all such molecules are active in UV visible region. And what is the UV visible region? It falls in the range of 200 to 400 is the UV, and from 400 to 800 nanometers is the visible range in which we do the particular study. Now, let us understand about the instrumentation of the UV visible spectrophotometer. The instrument has one important light source. When we talk about light source, the 2 light sources that are used are tungsten filament lamp and hydrogen deuterium lamp. The tungsten lamp has the ability to produce light radiation which are in the ultraviolet range, whereas hydrogen deuterium lamp produces light radiation for the visible range. So, the two type of light sources are clubbed together which act as the source for producing the desired light radiation of UV and the visible range. Then after the light source comes the monochromator. So, what is a monochromator? A monochromator is or it is an arrangement of a prism which has two slits into it. When the light radiation passes from the light source, it goes from the first slit and then pass through the prism. The prism, due to the process of diffraction, it produces the light radiations of the desired wavelength, which then passes through the second prism, slit, and comes out, and then it goes and fall on the beam splitter. So, this is the path that the light is following. The beam which is selected from the slit in the monochromator is actually getting divided. When it falls on the beam splitter, the splitter is that which divides its light radiation into two equal halves. Both the light parts will have same energy, same frequency, but they will only be divided so that they can simultaneously fall on two different compartments where we are keeping our sample and the reference, which is the next part of the instrument. So, let us see what is it. After monochromator comes the sample holder and the detector. So, the sample holder is made up of very specific materials. It is not made up of glass. What is a sample holder? These are small test tube like structures which are specifically known as cuvettes, and they are not made up of simple glass. Instead, they are made up of quartz. So, what is the reason that glass is not used and quartz is used? Because glass shows absorbance in the UV visible region. So, if we take it as a sample holder, the material of our holder will itself absorb the light radiation and the results will be varied. Therefore, quartz or silica is used for making these small cuvettes. If we can see the structure of the cuvette, these are small rectangular shape, three-dimensional holders, and the important thing about it is that two of the opposite surfaces of the cuvette are opaque, whereas two are transparent. So, when it is placed in the instrument, it has to be placed such that the transparent side falls in the region of the light path. And here in the diagram, we may see that there are two places of holders. One is the sample cell and other is the reference cell. So, this diagram is representing a dual nature of the UV visible spectrophotometer, or it is known as a double beam spectrophotometer. At a time, the beam has divided into two parts and it is falling on two cells. One is holding the reference and the other is holding the sample. So, after the sample holder comes the detector. The detector is what? Detector is actually a photocell which has the purpose, which detects what is the change in the light that has occurred after the light has passed through the sample. So, the photocell receives the beam from the sample cell and from the detector, and then it passes of the reference, and then the intensity of the radiation from the reference cell is compared with the sample cell, and finally we try to find out that what change has occurred in the energy of these particular samples. Then after the detector is the amplifier and the recorder. What is the use of amplifier? The amplifier itself, the term is suggesting amplify means to increase the intensity, because the detector has just detected the signals, the photoelectric signals which are coming. It has detected it, but might be these signals which have been produced are not very strong, so that they cannot be reproduced as some result and we cannot identify them. Therefore, we need amplifier to increase the intensity of the signals that have been produced by the photocells. So the alternating current which is generated is amplified, it's increased, and then this amplifier is connected with the recorder, and recorder is our final device which has a pen, and it produces the result in the form of a spectra. So after the detector, outside here is the recorder which has a pen-like structure, and when it moves it produces the graphs or the spectra, and these spectra are then finally interpreted, by which we come to know the specific information about the molecule. Now, if I am talking about the specific information, the UV visible spectroscopy, since we have said earlier that it causes electronic transition, so what is the result that is being interpreted from these spectra? We come to know the presence of conjugated bonds in the molecule. We must remember that UV visible spectroscopy is always used to study the conjugation in the molecule. So this is one important spectroscopic techniques which is used in various branches of sciences. In the next class, we will be studying the further use of UV visible spectroscopy. I hope you must have understood about the basic principle and instrumentation of this technique. Thank you.